Hi, welcome all. Yesterday, you all have learned already that what NGS data is and how you people will be analyzing the NGS data, how you will use in different field. This session will be just a spin-off of that. I will be giving you future aspect of NGS and what you people can do in different kind of fields using this NGS data analysis. So let's start the session. And if in between you find that I'm not audible or something, you can just ask, you can just tell. Okay, so this is word cloud, just to tell you that what all that things are that you can do while analyzing the NGS analysis. Some of the aspect is already covered yesterday, so I will not go into the detail, but just to tell you that just from the environmental sample to the plant sample and human disease sample, you can do anything. It's a vast field. Bioinformatics is a vast field, and NGS is useful in each and every field you can think of. So just a quick recap. In 1977, with the advent of Sanger method sequencing, the sequences as they start coming up, but those were the fragments of the sequence, not the full length sequences, not the whole genomes, but a specific part. And that too, if you don't know the sequence, it was very, you can say it was very difficult to just uh, amplify those reasons. So what scientists used to do is they used to make degenerate primers. The wet lab people might be knowing about the degenerate primers, and using that degenerate primers, they used to sequence their samples, and the samples were run on big gels, and you can just read ATGC nucleotides in that and try to form your sequence. And after that, in 1990, when Human Genome Project was kind of completed and announced that yeah, we can sequence the whole genome, the field actually took off, and everybody was behind this complete genome sequencing. And with that in mind, a lot of sequencers started to come in the market. So we have already seen yesterday's session that we have divided this era into three parts. First generation sequencing, which covers Sanger sequencing, then second generation sequencers are the sequencers we are using right now, along with the third generation sequencers like nanopore. You can see in the screen, it is just the size of a pen drive. So you can just go anywhere with this pen drive. You can just take the sample, mix it with the appropriate chemical, put it in this pen drive shaped nanopore, and run it in your computers or laptops. And you will get it will take anywhere from 3 hours to 6 hours, depending upon the sample, and you will get your sequences in a run. So right now, there are some issues with the uh, accuracy of the sequencing with nanopore, but the scientists are working towards it, and they have actually rectified that problem at a large scale. So now nanopore sequencing, I will not suggest to use it for variant calling, but yeah, for quick identification, like for surveillance during COVID times, nanopore sequencers were used very highly, along with the second generation sequencers and third generation sequencers like PacBio. So these third generation sequencers are able to give you a long read extending from 1 kb till 450 kb, 400 kb, and you can, even using nanopore sequencers, you can sequence the whole genome in a go. Either you convert that genome into DNA and sequence, or if you want to directly sequence RNA, you can do that also. And if you want to do metagenomics or transcriptomics or anything else, nanopore sequencers has the capability to do everything. And now this is just the DNA part. Now they are coming up with nanopore protein sequencers, where you can just do your proteomic work using the same thing. So actually the future is very bright, and it's exciting to see the development here.